All right, class. So in our last video, we did see how the different ions and how water gets out of the loop of Henle into the interstitial space. Now the question is, where will those materials go, right? Will they just hang out in the interstitial space of the medulla? Of course not, right? So I did mention that they will be picked up by the blood. Well, some of them. And before we go on to that, I really want you to understand how the blood flows through the vasa recta and which way the filtrate is moving. So let's draw this out. So at this point, what color is this? Let me see if I can change color. I'll use purple. It's a pretty color. So here we have glomerular filtration, right? So we have filtrate here. This filtrate will move down the proximal convoluted tubule, down the loop of Henle, and then it'll go through the distal convoluted tubule, right? And then ultimately get dumped into the collecting duct. So that's the pathway of the movement of the filtrate. How about the blood in the vasa recta? Which way does blood flow? I'll do this in red. So from the glomerulus, blood goes into the efferent arteriole, which then branches to form the vasa recta. So this is the direction of the blood flow. Do you see that? Why am I even showing this to you? I want you to look at the descending limb of the loop of Henle, which is this one, descending limb. And I want you to look at this segment of the vasa recta. Do you see that the fluids are moving in opposite direction? Do you see that? The fluids are moving in opposite direction. And same is true for this part in the ascending limb. The filtrate is moving in one direction and the blood is flowing in the opposite direction. Do you see that? Okay, so keep that in mind. And since they're moving in different direction, there is good exchange, and we will look into it in our next slide. So let's take a look here. So one more time, let's review the different parts. Oops, sorry. Let's review the different parts quickly. So this part is my proximal convoluted tubule. This is the descending. Why cannot I draw? This is the, I don't know. Okay, this is the descending limb. Then this is the ascending limb, right? Oh, now I can draw. So this is descending limb. This is all ascending limb. This is distal convoluted tubule, right? Okay. And then we also see the vasa recta. So now, so filtrate is going this way. Right? This is the way the filtrate is moving, and blood I just cannot get the color. Anyway, and blood is moving. I'll just use black for everything. I'm sorry. Blood is moving the opposite direction. So as blood comes down the vasa recta, the blood thinks, at this point, let's say blood is here. The blood thinks, so much solutes. Let me pull some in. So at this point, we have sodium, chloride, potassium getting into the blood. So we have sodium, chloride, potassium, sodium, chloride, potassium, all these things, all these solutes enter the blood. And at the same time, this is extremely annoying without Okay. And at the same time, water leaves. Okay. So at this point, the ions enter and water leaves. Okay. Now, thank God. I'll just draw blood in red arrows. Okay. Now, this blood, and as this happens, as it picks up more solute and gets rid of water, what is happening to the solute concentration in vasa recta? It's getting high, right? So at the bottom, it has high solute. Do you agree? A lot of solute and less water. Agreed? And then this will move up. As it moves up, what happens to the, think of the osmolality of the medulla. The medullary osmolarity goes down as the blood moves up, right? 900, 700, 500. So at this point, all the salts will be dumped out. So it will dump out salt, dump out sodium, chloride, potassium. It'll dump out sodium, chloride, potassium, and it will take up more water. And it'll take in water, water. Okay. Why is this thing important? Why? All right, class. So we will talk about the importance of this, but before that, let me tell you, the thing that I just showed you is called countercurrent exchanger mechanism. Why countercurrent? Because if you look at the proximal, if you look at the descending limb of the loop of Henle and the vasa recta, filtrate and blood is flowing in opposite direction. And the same is true if you look into the ascending limb and the vasa recta. Filtrate and blood is flowing in the opposite direction, right? What else did we learn? We are looking into the blood, right? As blood moves down, so we are looking here. As blood, so let me put this, as blood moves down the vasa recta, salt gets into the capillary and water gets out of the capillary, as shown by the black arrows and the blue arrows. That results in hypertonicity of blood or higher osmolality of blood. So blood has more solids and less water. However, as this blood moves back up the vasa recta, salt goes out and water comes in, as shown by the yellow arrows and the blue arrows respectively. And this is called the countercurrent exchange mechanism. Why is this important? This is really, really important to remove NaCl. Actually, this is really, really important to maintain the gradient of NaCl in the medullary interstitial space. Okay, now think about it. There can be a situation where, water can just sweep out all the salt from the medulla. So in order to prevent all the salt from getting flushed out of the medulla, the vasa recta actually pumps this, picks up the salt and then dumps out the salt in order to maintain the gradient. Another important reason why there is this blood flow is to provide oxygen. Remember, the vasa recta is carrying oxygenated blood. So it carries oxygen to the cells and tissues of the medulla. So these are the two most important things why we have this countercurrent exchange mechanism. If you go through your study guide, I will actually write all this down in writing so it's easier for you to follow. All right? Okay. All right, we will start with a new video.